This is a Chapter 1 summary of microbiology provided by OpenStax, which is free to everyone. Microorganisms, or microbes, are living organisms that are generally too small to be seen without a microscope. Throughout history, humans have used microbes to make fermented foods such as beer, bread, cheese, and wine. Long before the invention of the microscope, some people theorized that infections and diseases were spread by living things that were too small to be seen. They also correctly intuited certain principles regarding the spread of disease and immunity. Antony van Leeuwenhoek was the first person to describe observations of bacteria in 1675, the 17th century, using a microscope. During the golden age of microbiology, 1857 to 1914, microbiologists including Robert Koch and Louis Pasteur discovered many new connections between the fields of microbiology and medicine. Carolus Linnaeus developed a taxonomic system for categorizing organisms into related groups. Binomial nomenclature assigns organisms Latinized scientific names with a genus and species designation. A phylogenetic tree is a way of showing how different organisms are thought to be related to one another from an evolutionary standpoint. The first phylogenetic tree contained kingdoms for plants and animals, and Ernst Haeckel proposed adding a kingdom for protists. Robert Whittaker's tree contained 5 kingdoms: Animalia, Plantae, Prostida, Fungi, and Monera. Carl Woese used a small subunit ribosomal RNA to create a phylogenetic tree that groups organisms into 3 domains based on their genetic similarity. Berge's manuals of determinative and systemic bacteriology are the standard references for identifying and classifying bacteria respectively. Bacteria can be identified through biochemical tests, DNA, RNA analysis, and serological testing methods. Microorganisms are very diverse and are found in all 3 domains of life: archaea, bacteria, and eukarya. Archaea and bacteria are classified as prokaryotes because they lack a cellular nucleus. Archaea differ from bacteria in evolutionary history, genetics, metabolic pathways, and cell wall membrane composition. Archaea inhabit nearly every environment on Earth, but no archaea have been identified as human pathogens. Eukaryotes studied in microbiology include algae, protozoa, fungi, and helminths. Algae are plant-like organisms that can be either unicellular or multicellular and derive energy via photosynthesis. Protozoa are microcellular organisms with complex cell structures. Most are motile. Microscopic fungi include mold and yeast. Helminths are multicellular parasitic worms. They are included in the field of microbiology because their eggs and larvae are often microscopic. Viruses are acellular microorganisms that require a host to reproduce. The field of microbiology is extremely broad. Microbiologists typically specialize in one of many subfields, but all health professionals need a solid foundation in clinical microbiology. Okay, Chapter 2 summary of microbiology from OpenStax, which is free to everyone. Light waves interacting with materials may be reflected and absorbed or transmitted. Light waves can interact with each other, which is interference, or be distorted by interactions with small objects or openings, diffraction. Refraction occurs when light waves change speed and direction as they pass from one medium to another. Differences in the refraction indices of 2 materials determine the magnitude of the directional changes when light passes from one to the other. A lens is a medium with a curved surface that refracts and refocuses light to produce an image. Visible light is part of the electromagnetic spectrum. Light waves of different frequencies and wavelengths are distinguished as colors by the human eye. A prism can separate the colors of white light dispersion because different frequencies of light have different refractive indices for a given material. Fluorescent dyes and phosphorescent materials can effectively transform non-visible electromagnetic radiation into visible light. The power of a microscope can be described in terms of its magnification and resolution. Resolution can be increased by shortening wavelength, increasing the numerical aperture of the lens, or using stains that enhance contrast. Antony van Leeuwenhoek is credited with the first observation of microbes, including protists and bacteria, and simple microscopes that he made. Robert Hooke was the first to describe what we now call cells. Simple microscopes have a single lens, while compound microscopes have multiple lenses. Numerous types of microscope use various technologies to generate micrographs. Most are useful for a particular type of specimen or application. Light microscopy uses lenses to focus light on a specimen to produce an image. Commonly used light microscopes include bright field and dark field, phase contrast, differential interference contrast, fluorescence, confocal, and two-photon microscopes. Electron microscopy focuses electrons on the specimen using magnets, producing much greater magnification than light microscopy. The TEM, which is transmission electron microscope, and the SEM, scanning electron microscope, are two common forms. Scanning probe microscopy produces images of even greater magnification by measuring feedback from sharp probes that interact with the specimen. Probe microscopes include the STM, scanning tunneling microscope, and the AFM, atomic force microscope. Samples must be properly prepared for microscopy. This may involve staining, fixation, and/or cutting thin sections. A variety of staining techniques can be used with light microscopy: gram staining and acid-fast staining, endospore staining, and flagella staining. Samples for TEM, transmission electron microscope, require very thin sections, whereas samples for SEM, scanning electron microscope, require sputter coating. Preparation for fluorescence microscopy is similar to that for light microscopy, except that fluorochromes are used. Chapter 3 Summary from OpenStax Microbiology, which is free to everyone. The theory of spontaneous generation states that life arose from non-living matter. It was a long-held belief dating back to Aristotle and the ancient Greeks. Experimentation by Francesco Redi in the 17th century presented the first significant evidence refuting spontaneous generation by showing that flies must have access to meat for maggots to develop on the meat. Prominent scientists designed experiments and argued both in support, John Needham, and against spontaneous generation, Lazzaro Spallanzani. Louis Pasteur is credited with conclusively disproving the theory of spontaneous generation with his famous swan neck flask experiment. He subsequently proposed that life only comes from life, Latin, omni vivum ex vivo. Although cells were first observed in the 1660s by Robert Hooke, cell theory was not well accepted for another 200 years. The work of scientists such as Schleiden, Schwann, Rimac, and Virchow contributed to its acceptance. Endosymbiotic theory stated that mitochondria and chloroplast organelles found in many types of organisms have their origins in bacteria. Significant structural and genetic information support this theory. The miasma theory of disease was widely accepted until the 19th century, when it was replaced by germ theory of disease, thanks to the words of Semmelweis, Snow, Pasteur, Lister, Koch, and others. Prokaryotic cells differ from eukaryotic cells in that their genetic material is contained in a nucleoid rather than a membrane-bound nucleus. In addition, prokaryotic cells generally lack membrane-bound organelles. Prokaryotic cells of the same species typically share a similar morphology and cellular arrangement. Most prokaryotic cells have a cell wall that helps the organism maintain cellular morphology and protects it against changes in osmotic pressure. Outside of the nucleoid, prokaryotic cells may contain extrachromosomal DNA in plasmids. Prokaryotic ribosomes that are found in the cytoplasm have a size of 70S. Some prokaryotic cells have inclusions that store nutrients or chemicals for other uses. Some prokaryotic cells are able to form endospores through sporulation to survive in a dormant state when conditions are unfavorable. Endospores can germinate, transforming back into vegetative cells when conditions improve. In prokaryotic cells, the cell envelope includes a plasma membrane and usually a cell wall. Bacterial membranes are composed of phospholipids with integral or peripheral proteins. The fatty acid components of these phospholipids are ester-linked and often used to identify specific types of bacteria. The proteins serve a variety of functions, including transport, cell-to-cell communication, and sensing environmental conditions. Archaeal membranes are distinct in that they are composed of fatty acids that are ether-linked to phospholipids. Some molecules can move across the bacterial membrane by simple diffusion, but most large molecules must be actively transported through membrane structures using cellular energy. Prokaryotic cell walls may be composed of peptoglycan, bacteria, or pseudopeptoglycan, archaea. Gram-positive bacterial cells are characterized by a thick peptoglycan layer, whereas gram-negative bacterial cells are characterized by a thin peptoglycan layer surrounded by an outer membrane. Some prokaryotic cells produce glycocalyx coatings, such as capsules and slime layers that aid in attachment to surfaces and or evading host immune systems. Some prokaryotic cells have fimbriae or pili, filamentous appendages that aid in attachment to the surfaces. Pili are also used in the transfer of genetic material between cells. Some prokaryotic cells use one or more flagella to move through water. Patricious bacteria, which have numerous flagella, use runs or tumbles to move purposefully in the direction of a chemical attractant. Eukaryotic cells are defined by the presence of a nucleus coating the DNA or genome and bound by a nuclear membrane or nuclear envelope, composed of two lipid bilayers that regulate transport of materials into and out of the cell nucleus through nuclear pores. Eukaryotic cell morphologies vary greatly and may be maintained by various structures including the cytoskeleton, the cell membrane, and/or the cell wall. The nucleolus located in the nucleus of the eukaryotic cells is the site of ribosomal synthesis and the first stages of ribosome assembly. Eukaryotic cells contain 80S ribosomes in the rough endoplasmic reticulum, membrane-bound ribosomes, and cytoplasm, free ribosomes. They contain 70S ribosomes in the mitochondria and the chloroplasts. Eukaryotic cells have evolved an endomembrane system containing membrane-bound organelles involved in the transport. These include vesicles, the endoplasmic reticulum, and the Golgi apparatus. The smooth endoplasmic reticulum plays a role in lipid biosynthesis, carbohydrate metabolism, and detoxification of toxic compounds. The rough endoplasmic reticulum contains membrane-bound 80S ribosomes that synthesize proteins destined for the cell membrane. The Golgi apparatus processes proteins and lipids, typically through the addition of sugar molecules producing glycoproteins or glycolipids, components of the plasma membrane that are used in cell-to-cell communication. Lysosomes contain digestive enzymes that break down small particles and cells ingested by endocytosis, large particles or cells ingested by phagocytosis, and damage intracellular components. The cytoskeleton, composed of microfilaments, intermediate filaments, and microtubules, provides a structural support in eukaryotic cells and serves as a network for transport in intracellular materials. Centrosomes are microtubule organizing centers important in the formation of the mitotic spindle in mitosis. Mitochondria are the site of cellular respiration. They have two membranes, an outer membrane and an inner membrane with cristae. The mitochondrial matrix within the inner membrane contains the mitochondrial DNA, 70S ribosomes, and metabolic enzymes. The plasma membrane of eukaryotic cells is structurally similar to that found in prokaryotic cells, and membrane components move according to the fluid mosaic model. However, eukaryotic membranes contain sterols, which alter membrane fluidity, as well as glycoproteins and glycolipids, which helps the cell recognize other cells and infectious particles. In addition to active transport and passive transport, eukaryotic cell membranes can take material into the cell via endocytosis or expel matter from the cell via exocytosis. Cells of fungi, algae, plants, and some protists have a cell wall, whereas cells of animals and some protozoans have a sticky extracellular matrix that provides structural support and mediates cellular signaling. Eukaryotic flagella are structurally distinct from prokaryotic flagella but serve a similar purpose, locomotion. Cilia are structurally similar to eukaryotic flagella but shorter. They may be used for locomotion, feeding, or movement of extracellular particles. This was read by me, so I can concentrate on what on earth is microbiology. Thank you.